will be Feng, who is joining us from Ohio State University and the Department of Plant Pathology. And Feng will be talking about how viruses evict close relatives and why.
I cannot seem to figure out where to find the stuff. Yeah, okay. Thank you. Okay. Well, first of all, like all the speakers, I would like to thank YQ and also Jonathan for inviting me here. So it's a great opportunity, and I already learned a lot from the previous speakers, and I hope my talk will be of little value to others too. Okay, like Jonathan said, my name is Feng. I'm from Ohio State University Department of Pathology, and I'm a plant virologist. I think this audience is smart enough to know that plants also have viruses. So this is, first of all, I want to acknowledge the people who actually did the work. So I have a few graduate students in the lab, and on the top row, and then a technician, visiting scholars, and the last two are the former lab members, Feng and Ali. And Feng actually did most of the work that I'm going to talk about here. So the specific phenomenon here I want to talk about here is actually something called superinfection occlusion. What this is that when a cell is already invaded by a virus, this same cell will not allow the secondary invasion of the same virus. So the keyword is the same virus or very closely related virus, but it would not affect secondary infection by a more distantly related virus. So this is really a self-discrimination. So just like I like the example, it's just like your parents, they like pets, they can have 7 dogs, 8 cats, but they just wouldn't allow you to live in their basement. This is, so, and this actually was discovered a long, long time ago. And so here you are looking at the tobacco leaf. On one side of the leaf, it was first infected with a virus called tobacco black rain virus. It's a mild strain, so you don't see much symptoms. And so the bottom half is the same thing. You infected with the same first virus. But then 2 weeks later, on the top part, you reinfect with a more severe strain of the same virus. So the infection was prevented. And the lower half is with different virus. You can see symptoms. You can see infection. So what's more interesting, this is actually also conserved among animal viruses. A few examples include West Nile virus, dengue virus, hepatitis C virus, and so on. So here I'm using a very simple plant virus we call turnip crinkle virus as the example to study this phenomenon a little further. This is actually a virus with a single-stranded RNA genome. So it's we call it positive sense RNA virus. So this virus is very simple, only 4 kb genome, encoding only 5 proteins, the P28 and P80. These 2 proteins are the only protein needed for the virus to make more of themselves. It's called replication protein. The middle 2 are the movement proteins, and here this is capsid protein. So these proteins are not required for replication, so we can replace the capsid protein with GFP and mCherry so we can monitor them better at the cellular level. And so, we also use a more distant related virus, but really it's in the same family, have very similar coding strategy and so on. So this is used as control. As you can see in the next slide, when you infect cell with both modifier from TCV in this row of cell, I cannot seem to use the mouse to point. So this is a little. You can see on the left-hand row that if you co-infect plant cells with the, M. cherry and GFP labeled, but the same virus, they do not. So you have roughly same number of cells infected, but they never coexist in the same cell. So this exclusion occurs at the cellular level, even if you deliver both at the same time into the cell. So, but if you use TCV and another virus, carnation model virus, they live in the same cells very happily. So what this says is that this is indeed self-discrimination, and that you can introduce both of them in the same cell at the same time. They still occur. So this is just a summary. So what caused them to exclude each other? So the next few slides, I will save you all the experimental details because most of you are not virologists, may not be. But I want to highlight that this phenomenon, this superinfection occlusion, is actually conferred by the P28, the replication protein of this virus. So what this tells you is that the replication protein facilitates the replication of the primary TCV, but represses the replication of super-infecting TCV. And also, we found that this is through a P28 interaction. So all the details were published, so I'm not going to talk about too much. Instead, I'm going to extend from this results and talk about some hypotheses. So how does, the key question is that by the time of the superinfector you get in touch with the cell, the primary virus would have made thousands, if not millions, of progeny viruses. So how can the cell can tell which one is progeny, which one is superinfector? So the logical conclusion, or rather our hypothesis, is that this P28-mediated repression does not specifically target P28 or translate from superinfector. Rather, it actually targets all P28, probably in a concentration-dependent manner. And actually, the intended target of this superinfection occlusion are indeed the progeny viruses, just like your parents. They don't want you to reproduce in their house. So why would virus do that? So yeah, here's our model I want to go. So for those of you not familiar with virus replication, it's typically the type of virus we work on, like TCV, they replicate on some kind of cellular membrane. And typically the protein, in this case P28, through a so-called nucleated polymerization, they form a two-dimensional lattice on the top, on the surface of membrane, and then cause membrane to bend, and eventually form this kind of, we call it spheral. And inside the spheral is where virus replication occurs. So being able to undergo nucleated polymerization is the innate ability or property of this protein. However, when the replication gets started, then you produce tons of progenic RNA, and those progenic RNA can serve as template for the translation of a huge amount of P28. And then if you have too many P28, this polymerization process becomes so fast, the kinetic becomes so fast, so the membrane would no longer able to enclose them, and they stay on the surface. And furthermore, because they still retain the polymerization property, they keep the ability to attract and trap new monomers to this, so they prevent newly translated P28 from getting involved in new replication. So this is our model how it gets the replication of the progenic virus RNA gets stopped through this kind of trap. Furthermore, we think we're going to test that RNA actually can even get trapped in this complex as well. So again, I promised to tell you why would this happen. So here is one model that because RNA virus replication is an error-prone process, on average for our virus, they would introduce one error per genome per replication round. So as you can see here, this is a hypothetical situation. So round one, you have this random mutation. Those random mutations are good because they create a pool of variations for adaptation. However, if you allow those to go another round, and then some of the mutation would get spread to many more progenic RNAs, and plus it would be superimposed by additional mutations. And this process go on and on, then this genome would become less and less fit. So there's a need, evolutionary need to control this mutation rate. But there's even more to that. Actually, this becomes less important when you see next example. And this example was published in 2015, was not our work, was by somebody else. But this is interesting in that what they did is create a mixture of virus genome by randomizing this terminal. So in their case, they estimated each cell, this is protoplasm cell, could get up to 5,000 different genomes. They could identify them through high-throughput sequencing. And then they sequenced 15 individual cells. What they found is that despite this huge amount of input, each cell would replicate only up to 7 different genomes, and not to even, it's not evenly distributed. For example, some of them only very small percentage. So there's a very severe intracellular population bottleneck. So why is that? So here is, I think our model can explain that. So if in a plant infected by virus, even though the first cell might only encounter a few viruses, but the majority of cells actually encounter large amounts of viruses. They get virus from neighboring cell. This is shown here in this diagram. So this virus, this is first cell invaded by virus, and they produce this, so in our case, P28 protein, because if it's one virus, you produce very little, a small amount of the P28 protein. So most of the virus could replicate, and because this P28 is not enough to build up the repressive structure, so they will go on for a few rounds to make a lot of virus particles. But the subsequent cells, they each would get a lot of viruses and also a lot of genomes, of course. And these genomes then all translate to quickly build up this pool of P28. So in these cells, at the very beginning, the repressive state is already built. And this partially repressive state would allow only very few genomes to go on to replicate new progenies. So why this is important? Why this is important? Because this creates a situation to quickly purge the mutated, the defective genome. So say, for example, in this cell, this genome randomly, this is a random genome that gets replicated. This is not defective, so the go on to replicate. But in this side of cell, it is the mutant genome. Because there are so many other RNA making this proteins, so there is a chance this mutated genome could be replicated and making mutant virus. But then in the next cell, because they make so many of them, so the next cell would only get the mutated genome, mutated virus. And this gets quickly purged out of the population. So with that, I think this is pretty much my last slide. And suggestions and criticism would be highly welcome. And if I'm looking for collaborators. And finally, thank you all very much.
So are you suggesting now we should immunize crops?
What's that?
So, I mean, where this work leads to a suggestion, say, crops can be immunized using a virus for certain viral disease.
Well, yeah, in theory, if you make transgenic plants using the P28 protein, it expressed to high concentration, yeah, it could, in theory. That's how crop protection works. Crop protection is you use milder to treat the plant, prevent the secondary effects. But in reality, because this protein is highly depolymerized and they form protein aggregate, it's like crayon, to be honest with you. So it often becomes detrimental to plants. So transgenic plants, maybe you can make inducible, you do inducible expression, it might work. But, you know, we did that, but the plants we recover, we express very low level of the protein. Yes.
Thank you. It was a very interesting talk. So, in this phenomena, only the protein, the P28, is needed and it is sufficient for this phenomena?
For the
Suppression, yes.
Our case, P28 is enough. However, you can see that I probably forgot to explain. In our particular virus, the P88 is actually what we call it a read-through of the P28 stop product. So if you overexpress P88, you actually get the same effect. Yeah.
Okay, thank you.
All right, let's thank Feng. Yep, and that concludes our first session. So you can now grab some coffee. We'll have a quick coffee break before the second session.